Good morning, everyone. Thank you for coming to my session, and thank you for the warm introduction. It is my pleasure to be here to introduce our work on intergeneration transmission of mental illness. I'm going to particularly focus on prenatal factors as well as genetics, how they are influence the early brain development. So I have no disclosures. As shown in this chart that published by WHO, you can see the prevalence rate of mental illness start to rise from age 5 onwards. There's little knowledge about clinical determinants as well as diagnosis clarity of mental illness in children before age of 4. And we really think this is a big gap. We should understand the developmental origins of mental illness. And we face two challenges. The first challenge is that we are nowhere close to understanding environmental factors and biological processes that shape brain development in early life, which may lead to mental illness in later life. The second, we don't know how to predict which child will develop a mental illness, which form of the illness will take, and what to do to prevent such fate. So we propose a theoretical fetal programming model to enhance our understanding and how this, environmental factors as well as biological processes that can shape early brain development. The fetus basically stays in intrauterine environments, and the environments are influenced by maternal exposure to external conditions. The maternal environment during pregnancy basically refers to a very complex biological, psychological, and social conditions. And this includes, for example, socioeconomic status, psychological distress such as depression, anxiety, physical health problems such as metabolic disorders and nutrition problems and so on, as well as lifestyles. And of course, all these things influences intrauterine environment and basically influencing the fetal brain development as well as sets the foundation for long-term health problems. So biologically, how that can happen, and we basically, there's central to the transmission of these maternal effects is mainly related to a placenta. And placenta plays a vital role and basically, that connects the communication between a mother and child. And alteration in placenta functions that may be influences by the intrauterine environment, and that can mediate a lot of biological pathways, such as something related to endocrine systems, metabolic systems, as well as the inflammatory systems. And certainly, and this molecular information can be remembered by the fetus. And such that they will be able to, they will memorize in our biological system that influences the future neurodevelopment outcomes. Moreover, we also, child born with nature, right? Some of the child are more vulnerable to maternal environment factors, but some are not. And those genetic vulnerability probably most relevant to the biological systems, such as stress system and hypothalamus pituitary adrenal HPA axis, as well as neurotransmitter systems and neuroinflammatory systems that mainly influences brain development. So in this talk, I basically going to explore this model using the longitudinal studies we have built since 2009. And this longitudinal study is called GUSTO, Growing Up in Singapore Towards Healthy Outcomes. The GUSTO study is very much designed in epidemiology manner. So we recruited about 1,200 pregnant women during pregnancy. And they are actually a population representative in Singapore. And we actually study a deep phenotype, a lot of maternal factors, such as psychological distress, diet, lifestyle, socioeconomic status, family support, and pregnancy complications, as well as biospecimens that allow us to study biological systems. So the major outcomes of the study is mainly focused on children, spanning the measurements related to birth outcomes, metabolic problems, and brain imaging, cognition. We also have a lot of biospecimens that allow us to study epigenetic information of the child. For specimen of mother, we collect the blood samples and buccal swab during the second semester of the pregnancy. We also have placenta tissues at birth, and breast milk was acquired during the postnatal period. And for children, we collect biospecimens at multiple time points, including cord blood, stool, and buccal swab, as well as blood samples. To just give you an overall view what we acquired for neurodevelopmental assessments. At infancy, we actually conduct a first experiment, facial imitation in the first 24 hours of life, and in order to examine the emotion processes of infants. So the first MR scan was done in the second week after birth. At 6 and 18 months, we use a variety of technology in order to test child memory function as well as executive functioning and mother-child interactions. The second MRI scans was done in the 6 months of age. So that was pretty hard. So we only did a very small sample at the 6 months of age. From 2 years onwards, we basically examine executive functioning, memory, emotion, share of mind. And at 2 years of age, we use Bailey scales, child behavior checklist, CBCL, to examine global cognition, motor skills, as well as behavior of toddlers. And at 4 years of age, we use child diagnosis interview, skill, CDISC, to examine psychopathology of children. For academic, performance, we basically examine the, language function as well as numeracy using school readiness test. We also have IQ test as normal because everybody has to do it, right? So we also examine mother-child interaction as well as parenting style using Etch-a-Sketch as well as adulthood attachment. So we restart brain imaging at 4 years onwards, so we have multiple time points for, for children at age of 4, 6, 7, and 10 years, and so on. So now the kids are already 13 years old. To give you a brief view, what does the infant brain look like, and we actually generate the diffusion tensor imaging, DTI, as well as structural brain MR atlases using a probabilistic estimation approach. And this approach basically integrates both DTI imaging as well as structural imaging together in order to get a concurrent anatomy of the white matter and green matter. So the top 2 rows shows the color map of diffusion tensor imaging as well as fractional anisotropy map. And the last row shows the structural T2 weighted image for the neonates as well as 6 months olds. So you can see from this figure, and the brain grows dramatically in the first 6 months, but also white matter becomes much more coherent at 6 months of age. Nevertheless, imaging the infant brain is pretty challenging. And as you can see the last row, we can now clearly see the contrast between white matter and green matter. And this is mainly because the 2 tissues have very similar T1 and T2 values. In the traditional T1 and T2 weighted image, it's pretty hard to be able to distinguish white matter and green matter at this age. By age of 4, you can see the figures on the right. And you can see the brain images are very much like what you see in the adult brain. And again, we start to switch from T2 weighted image to T1 weighted image. So we then integrate the resting state functional MR in order to pathologize the whole brain. And for infants, the whole brain can be pathologied into 19 functional networks and 60 white matter tracks. And from 4 years onwards, the brain can be pathologied into 20 functional networks as well as 70 white matter tracks. And at least from what we observed down here, 4 years and 6 years and 7 years brains have very similar brain pathologizations in terms of functional organization as well as white matter information. So in collaboration with 6 other cohorts, we basically detail the brain growth of boys and girls in the first 2,000 days. And this can be become a standard reference for early brain development. And I will be, in the following of the talk, I'm going to use this data set from Gusto and to first explore whether the brain of neonates born to mother with depression look like the brain of depressive adults, especially in the cortical limbic circuitry. Does prenatal depression have a long-lasting effects on child neurodevelopment? So here, we particularly use neonatal brain imaging as surrogate markers for fetal brain development, because it's really not much of postnatal life influence. I think it's a very good model to be able to examine what happens in the uterine, during the uterine stage. So here, what we see is actually the greater the prenatal maternal depressive symptoms had a lower, the neonates born to mother with greater depressive symptoms had a lower fractional anisotropy in the right amygdala at birth. Functionally, we basically see an increased functional connectivity of amygdala with ventral medial prefrontal cortex, orbital frontal cortex, and insula. That's most relevant to emotion regulation networks. And we also see increased functional connectivity of amygdala with superior temporal cortex and medial temporal cortex, as well as entorhinal cortex that's more related to perception and memory functions. And structurally, we basically use amygdala volume as well as cortical thickness and study the structural covariance. And what we found again, and the neonates born to mother, with a greater maternal depressive symptoms, they actually have stronger amygdala and frontal structural connectivities. So when we look at 4 years and 6 years of a brain, and even after control a lot of postnatal factors, such as postnatal maternal mood, social economic status, and other factors, parenting styles, what we found is actually a greater maternal depressive symptoms is associated with a greater amygdala volume. That's particularly found in girls. And we then see a reduced functional connectivity of amygdala with medial prefrontal cortex, as well as orbital frontal cortex, and superior temporal cortex as well. And again, structurally, we see a reduced structural connectivity between amygdala and prefrontal cortex. And this pattern is really opposite to what we observe in the infancy, as I show you down here. And this may reflect a developmental shift, potentially something related to the role of amygdala from bottom-up processes, to top-down processes through the development during the early life. So what about other psychological distress factors? And we really want to know what other dimensions of psychological distress factors are the influences of fetal brain development. So what we do is here, we basically pull all the questionnaire items related to maternal mood during pregnancy. Then we conduct a p-factor analysis, and we found one general maternal mood factors along with 4 other subdimensions, including depression, anxiety, somatic symptoms, positive emotion, pregnancy-specific reactions. And this multifaceted patterns of psychological distress was validated using 3 cohort data, including Generation R, Gusto, and Mavens. So using this data, we basically found that the prenatal maternal anxiety slowed down the hippocampus growth in the first 6 months of life. And interestingly, greater positive emotion during pregnancy was associated with a larger hippocampus volume, particularly in girls. Additionally, we observe more effective suppression of functional connectivity among task-selective networks, including default mode, as well as the salience network, as well as enhanced connectivity among task-positive network, including RACH, frontal parietal network, visual associated cortices, thalamus hippocampus regions. And notably, the majority of these patterns are in opposite directions of those typically observed in children with anxiety and depression, suggesting that they may reflect key neural mechanism underlying the transgeneration transmission of maternal positive mental health to better children's brain development. Altogether, these findings suggest that neural substrates, particularly within the cortical limbic circuitry, may begin to develop in utero in response to maternal psychological distress. Moreover, maternal psychological distress exerts lasting effects on neurodevelopment. How that biologically happen? We like to ask how children's body memorize what happens previously, and how does that affect subsequent health. So as mentioned before, the Gusto phenotype allowed a variety of environmental measures during pregnancy, including maternal depression, and anxiety, socioeconomic status, lifestyle, and diet. So we have 2 key questions. One is, to what extent is prenatal environment reflected in neonatal mesothelium? The second, are these epigenetic signature associated with neonatal brain as well as children behavior later in life? So in Gusto study, we use cord blood samples and sequence the DNA methylation as well as the genotypes. We employ 3 linear regression models to examine the contribution of prenatal environmental factors, genetic variants, as well as G by E interactions on epigenetic markers. So what we found is that approximately 25% of epigenetic markers are best explained by genetic variants. The rest of 75% of genetic markers are best explained by G by E interactions. None of the epigenetic markers are explained by environmental factors alone. And to give you an illustrative example, and this scatter plot shows the association between prenatal maternal depression and DNA methylation was strong in one genotype, but weaker in the other 2. We further looking into a specific gene, brain-derived neurotrophic factor, BDNF. Its polymorphic RX6265 alters BDNF levels. Its variants are associated with differential response to adverse life events. From this figure, you can see the number of CPG sites correlating with prenatal maternal anxiety is disproportionately high in the neonates. When we stratify the neonates based on maternal genotypes, and this was not found. And this suggests that it is a neonatal, not mother, BDNF genetics that modulates epigenetic sensitivity to maternal anxiety. Furthermore, the maternal mesosome associated with a right amygdala volume in MAD neonates, but left hippocampus volume for VAD1 neonates. And this is very interesting, and this finding suggests that neonates with different BDNF genotypes react differently to maternal anxiety, and their epigenome reflects a variation in different brain regions. When we dive into an individual CPG, and we found this particular CPG sites lying between a BDNF antisense transcript and the long intragenetic non-coding RNA 678. And this particular gene methylation is associated with prenatal maternal mental health and predict child behavior at 3 years of age. So these findings actually suggest that the neonatal epigenome encodes a molecular memory of exposure to prenatal environment, shaped not only by environmental exposure alone, but predominantly through the interaction with the infant genetic makeup. And this work underscores the importance of considering both genetic and environmental contributions, and critically, their interactions. The fetal brain development is a dynamic and highly sensitive process shaped by intrauterine environment conditions. The offspring's genetic makeup likely play a role in determining their vulnerability. And such genetic vulnerability to prenatal influences is likely manipulated through a key biological pathways related to hypothalamus, pituitary, adrenal HPA axis, neurotransmitter systems, as well as neuroinflammatory response. And this system plays a central role in regulating neurodevelopmental processes during gestation and may determine to extent to which prenatal environment impact long-term brain outcomes. In order to examine the genetic vulnerability, and it's very, very challenging because the gastro samples are relatively small to study gene and environmental interactions. In order to achieve this, we basically develop a statistical model called the mixed effect model for gene size and environmental interaction. And we build this model mainly because, number one, the brain development is not driven by single gene. And second, the mental health problems is not driven by single genes, as in single SNPs as well. So what we do is try to study a set of SNPs, not just a single one of them, and estimate the overall interactions of those SNPs with prenatal factors. And in addition, we're also considering the heterogeneous of interactions across all these SNPs. So this model actually can handle a small sample size in order to study G by E interactions. First, we're going to use this model in the exploratory analysis way. So how I do it in the exploratory way? So what we do is actually we take the SNPs derived from existing GWAS study for depression, and looking at this set of SNPs interact with a prenatal maternal depression. So what we found is that the neonates with a higher genetic risk for depression and shows a greater thickness of ventral medial prefrontal cortex as well as orbital frontal cortex in the context of elevated prenatal maternal depressive symptoms. So likewise, we see the larger the hippocampus volume as well as the amygdala volume, and particularly in the hippocampus, we see the larger volume occurs in the anterior hippocampus that was detected using a shape analysis. When we go deeper, so we ask, among those SNPs, if we're looking into an individual subset that has a particular function, and how this contributes to such interaction, and we found the genes highlighted in yellow down here in this graph that's most relevant to glutamine receptor activity contributes most to the interaction on amygdala volume. And glutamate is a transmitter that plays many roles in our body, and many approaches, including GWAS study, has implicated its synapse transmission. Basically, its synapse transmission is in the etiology of major depressive disorder. And of course, its expression in amygdala actually has different levels between the controls versus a patient with MDD. And to basically validate our results, remember the gastro sample is small. We only have about 200 neonate imaging scans. For the above results I show, it's only conducted in 160 Asian mother-child and diets. So we validated this above results using a Caucasian sample contributed by UNC group. And you can see we can replicate the interaction effects as well as we can identify glutamate receptor activity as a major contribution to such interactions. However, if you look in depth, and these two figures shows the opposite interaction effects. And this phenomena has previously reported in other genes, and such as serotonin. And these results really suggest that the gene-environmental interactions influencing early development may be a population-specific, potentially shaped by genetic backgrounds. So next, we're going to use a hypothesis-driven way to explore the HPA axis as well as the immune system using the mixed effects, gene size, and inversion in the model. So the chronic stress appears to have long-term effects that contributes to immune dysregulation, promoting pro-inflammatory cytokine response. And stress can activate the HPA axis that stimulates the pituitary gland, leading to a release of adrenocorticotropic hormone, which in turn to increase cortisol, glucocorticoid production from adrenal glands. Glucocorticoid are the final effector of HPA axis and play a crucial role in regulating both immune systems and stress response. So in our study, we first take a FKBP5 gene, and it inhibits glucocorticoid receptor activity, and is a major regulator of HPA axis. And if FKBP5 has been identified as molecular genetic markers associated with major depressive disorder and post-traumatic stress disorder, PTSD. So in this study, we particularly take all the SNPs from this gene, and we use the mixed effects model to look at the interactions with prenatal maternal depression. So what is showing in this figure, you can see that neonates with higher genetic risk, they have a larger hippocampus volume in the context of greater prenatal maternal depressive symptoms. Furthermore, when we look at cytokines, and these are the major immune signaling proteins, and they can be sensing intrauterine environment, but also manipulates the fetal brain development. The cytokines are actually expressed in the brain in different brain regions. Then the fetus stage is very special stage of life. You can see down here, the gene expression information in the fetus stage is very different from those from early childhood as well as adolescence and adulthood. So in other words, some cytokine may be functional during fetus stage, but some cytokines may not. So what we do is actually we use brain span data to identify which cytokines are functional and which other brain regions they are functional during the fetus stage. So in this figure, you see about 25 cytokines are expressed during fetal stage at different brain regions. So we then use our model, mixed effects model, to looking at these functional cytokines interact with prenatal maternal depression. So what you see in this figure, again, for the neonates with higher expressed levels, and it basically shows larger hippocampus volume as well as the thicker orbitofrontal cortex. So all these findings suggest that fetal brain development is sensitive to the maternal environment, with vulnerability manipulated by genetic variation in HPA axis, neurotransmitter pathways, as well as inflammatory responses. So in the last few minutes, I'm going to talk to you about the second challenge I mentioned at the beginning of the talk. So how to predict the child behavior and psychopathology. So here we recently developed a multimodal deep learning framework for predicting behavior and psychopathology across the first 21 years. So this framework integrates the 3 cohort data, including GASTO, ABCD, and PNC. The total sample we include is more than 10,000 samples down here. And in this framework, we integrate the text information as well as multimodal brain images. And from brain images, we include the features of morphology, such as thickness, surface area, volume information, and for the functional information and DTI information, we include the function connectivity and structural connectivity. So for the text features, we particularly include information related to demographics, such as age, gender, race, parent education, and so on. Developmental adversity, and such as sleep disturbance, trauma experience, and so on. And parent psychopathology, as well as family and school environments. So we evaluated this model using area under ROC curve. What you see on the top is the AOC curves, and for the prediction accuracy for preschoolers, schoolers, early adolescents, as well as adolescents. You can see AOC values reach about 0.63 for preschoolers and increases to about 0.8 for adolescents. So based on the feature extraction and using the attention mechanisms in the deep learning framework, we found for preschoolers and early school, preschoolers and schoolers, maternal depression as well as sleep quality are the major contributions to the prediction accuracy. And for adolescents, and we see pro-social skills, sleep quality, as well as parents' psychopathology, that they are contributes most to the prediction. From brain imaging data, you can see on the top, these are morphology information based on cortical thickness, surface area, as well as volume. And the second row shows the subcortical volume contribution. What you can see down here, the brain regions, particularly in the cortical regions, that are most aligned in the unimodal axis, such as sensory regions, as well as the visual associated cortices, and those are the regions contributes most to the prediction. And this is actually consistent in the first 21 years of life. For the subcortical regions, the features are pretty much stable. That includes the cerebellum information, cerebellum volume, as well as basal ganglia volume. For functional connectivity and structural connectivity, you basically see the frontal parietal, DMN, as well as the cerebellum networks are the top 3 contributors to the prediction. So this is just give us a possibility that we were probably able to predict child behavior as well as psychopathology in early life. So we are still validating this model in order to make it more robust so that they can be a really good tools for us to be able to use to predict the children, particularly in younger age. And to just give a summary, and today I discuss a fetal programming model using our longitudinal data. And all our findings down here is based on very simple analysis, which is association analysis. Unfortunately, the systems, the body system is very complex, and certainly we are very much simplify what we can do at the moment. So basically, we are just looking at association. So in order to model these complex systems, I think we still need a lot more efforts, much larger samples, in order to really dig into the interactions, for example, the interactions among the environmental factors that we list down here. So what is really like in this talk as a full picture, as I mentioned before, is really related to the placenta functions, which is something we're going to look into in near future. If you like to understand this topic, please visit our posters. We do have some preliminary results related to this information, and come to talk to us this afternoon. Finally, I'd like to thank all my collaborators, especially my students and postdoc fellows, who actually contribute to a lot of analysis results I presented here today. Of course, we always have to thank our funding bodies. Without them, we couldn't conduct this complex analysis. But I also like to thank my institutions, previous institutions, National University of Singapore, as well as my current institution, Hong Kong Polytechnic University, to support my group in the great efforts. Thank you. Thank you for your attention.
Thank Dr. Qiu for a wonderful presentation covering so many different aspects of the factors and mechanisms contributing to the fetal development. So we have quite some questions from the audience. By the way, you can still submit your questions if you want. So the top one is related to your gene and environmental interaction study. So the audience asked, how can you distinguish prenatal environmental effects from heritability, basically, so the traits that could come from the mother directly?
For the gene interaction results I showed down here are pretty much focused on neonatal brain. One thing is actually they basically get rid of postnatal inferences. But in terms of heritability, we basically, some of our analysis really control the maternal genotypes as well, so maternal genetic variants as well. So in some way, we may be able to get, remove some of those information, but, certainly, we can answer for sure.
Okay, in a somewhat similar context, an audience also asked, wait, where is it? The epigenetic question. Oh, someone asked, did you examine twins in investigating epigenetic modifications?
Because our skeletal sample is relatively small, we do have some twin data, but very, very small portion. So we normally exclude them from the study. So in any analysis to reduce the complications. So those are, not twin data.
All right, a question from an audience for another study. Could you speculate on why there was a relationship between hippocampal volume changes and mental health, specifically in female? Is there a hypothesized underlying hormonal contributions to hippocampal volume difference?
That's actually a very good question. So I did not emphasize a lot of things related to sex. And the males and females and boys and girls, and this is particularly number one in neonatal study, and majority of findings we have that does not show gender sex difference. And so then later on, in at 4 years stage onwards, we basically see majority of findings actually is in girls, and this is probably mainly because the girls are more sensitive to emotion processes and so on. But when this sex difference occurs, and a lot of hypotheses says it should start from fetal stage. And I guess our samples, we only have about 200 neonatal brain images, so we will not be able to really see the differences between, girls and boys at the, birth. But it doesn't mean it doesn't exist. So, and this is something, I try not to, I didn't emphasize in my talk, so I hope, some of you guys have similar data. You can come together to see when this really happens. Yeah.
Still on hippocampal volumes, could you share your thoughts on what could be the behavior implications of the enlarged hippocampus of the children with mother at risk?
So far, we did not look into so much of behavior and brain associations. But you can see in the talk, based on morphological findings, we always see larger amygdala, larger hippocampus volume, as well as thicker the orbital frontal cortex at neonatal stage in response to a maternal depressive symptoms. And this actually, remember, our sample is pretty much studies epidemiology design, so it's actually a general population. We do have some mothers in extreme case, but a majority of mothers are still in the clinically normal range. And so, in other words, there's a model related to the stress response, because that stress level is relatively low in our sample, and it could trigger the, a brain development in early life. That could happen in that way. But when they go to severity, more severe cases, that may be a different story. So again, and when combining with large clinical samples, that particular like a case control studies with our epigenetic samples, and that probably give us a full spectrum what's happening down here.
And another related question is, were you able to measure antidepressant use in mothers, and did this change the impact on infants' brain?
So we do have some records related to the medicine, right? So the medication, right? So very, very few mothers take a medication. And so, and this is something we exclude from our study. So basically, we didn't look into the medication per se. And especially in Asian culture, there's actually a big concept related to not taking any medication during pregnancy. So that's something very different from maybe from other populations and so on.
And there are two questions about the data set. So Gusto is a really awesome data set. So someone asked, is it open for access?
So the GASTO has a management committee. It's called the executive committee. So everything has to be reviewed through it. So there's certain procedures you have to go through in order to access the data. Yeah.
And also there are two similar large data sets in the US, ABCD and PNC, but they do not fully overlap in their age range. So the audience wants to know your thoughts about how to resolve the ethnicity and culture difference there across different data sets.
I hope in the future we can do all of that, based on the Asian samples and Caucasian samples. As I showed down here, the genetic makeup is very different between, among populations. And I think that would be a very interesting topic. I hope that would be, we'll be able to answer. And I think wonderful results that I show, particularly replication examples, right? I show one from our sample, the other one from UCI sample. And we really see the interaction effects. However, the directions opposite to each other. And so the reviewers give us a really hard time during the time. So basically, they say you cannot say it's replication because you didn't replicate the same direction and so on. So, but we gave a lot of examples like showing in literature. And as I mentioned, serotonin is a big one, and so on. So what I expect that, there may be the same loci that plays the role, but the role is really depends on the genetic variants that up onto individual, ethnic groups and so on and so forth. So I think this is something still, on the year. So I guess if people like to study cross-cultural, cross-ethnic groups, I think this will be very interesting topic to look into.
Okay, I'm going to take the very last question. I know there's a lot more interesting ones, but this is more personal-ish, and I'm very curious to hear your answer too. So the audience asked, your research career seems to have moved from method-oriented to more clinically focused. What motivated this, and how do you see the two areas inform each other?
I'm an engineer. I trained as an engineer, so I still claim myself as an engineer. So I have two hats. One hat is more in the neuroscience research, the other hat is more in methodology. So in this meeting, most of the time I wear the hats of neuroscience relevant, clinical relevant research. But in MICAI conference and so on and so forth, we actually present methodology. So my group actually do have a lot of people who are working on methodology development. As I show you down here, basically, for example, stats model I introduced down here is developed by our own group, and the code is available online in GitHub. So quite a number of codes we developed is available on GitHub, so you can actually download it. And I think it's really interesting. I always develop methodology based on the clinical or neuroscience relevant questions. So we don't develop methodology just for that methodology. Basically, my group of people who develop methodology, the other people who are in background of psychology or neuroscience and so on, clinical background, they're going to use them. So we normally, integrate the two groups very well. So.
Okay, thank you very much. So before I close the session, I want to remind the audience, please provide your review in the app. That will help us and help the speaker to shape their talk in the future. And also I encourage those whose questions were not addressed, please come up and have a chat with Dr. Qiu. Thank you all for coming, and thank Dr. Qiu again.
Thank you very much.